Do you like looking at the stars? The night sky has always inspired mankind to question its existence. Where do we come from? What is out there waiting for us? People want to reach for the stars, explore new worlds, but they often forget the beauty of nature surrounding us down here on Earth. But how could this disparity emerge? In the beginning, there was only darkness. But with light came life. Light is the most abundant and reliable energy source of our planet, causing flowers to blossom and bloom, trees to grow, and providing animals with nutrients. Mankind has always relied on nature to provide us with food or medicine, leading to our survival. Technological advances have improved our way of living. Diseases have lost their danger, but simultaneously we seem to have forgotten how to live in harmony with nature. If we don't change our way of living, our survival is at stake. Facing climate change and scarcity of resources, finding more sustainable ways to produce is one of the most urgent goals of our society. Nature is fascinating. Nature can inspire us and show us innovative solutions. This is where our iGEM project, ET, the Electron Transferrestrial, comes into play. By using microbial fermentation for the production of important chemicals, we will become independent from the petrol industry and even make new products accessible. But achieving this is not without challenges. Fermentation in many cases leads to low titers of the desired products, and complicated extraction procedures are necessary. But fortunately, these drawbacks can be tackled by delicate metabolic engineering. Fermentation seems to be most suitable for the production of scarce, chiral, natural products with a high market value, in which case lower product titers can be justified and are still economical. We started our iGEM journey looking for the optimal product for our project. Terpenes and terpenoids appear to be a promising class of natural products for our requirements. Many terpenes are high-value products and are used as drugs as well as fragrances and flavors. There is a large market and great commercial interest in these substances, and other iGEM teams have worked with terpenes before. Moreover, once established in an organism, terpene biosynthesis offers a variable platform, since the synthesis of all terpenes starts at identical building blocks. Another important feature is that the metabolic pathways, mainly the mevalonate pathway, involved in this process are known. Additionally, the heterologous expression of required enzymes leading to the production of intermediates was successful in various organisms. Commonly, these intermediates need to be functionalized to obtain biological activity. For the functionalization, mainly cyclases and cytochrome P450, or CYP, monooxygenases are needed. In general, monooxygenases are enzymes that introduce an oxygen atom into a substrate, using NADPH as an electron carrier. The common feature of all CYP enzymes is that the electron transfer between the CYP enzyme and the final reductant often involves a cytochrome P450 reductase, or CPR, as an electron mediator. Natural fusion proteins of CYP enzyme and reductase show remarkably high turnover rates, since electrons can be directly channeled to the reactive site of the CYP. For our project, we therefore envisioned linking truncated versions of CYPs with appropriate CPRs to make the electron transfer more effective and optimize the metabolic pathway, which could lead to increased product titers. Additionally, we aimed to standardize the design of these fusion proteins and applied bioinformatics to develop a tool for optimizing the linker sequences between the CYP and CPR subunit. An optimal linker can significantly increase the performance of the fusion construct and should make the design of a variety of different CYP-CPR fusion constructs possible. We then looked into which terpenoids we wanted to produce. Artemisinin is a sesquiterpene lactone from the plant Artemisia annua and has proven to be a very efficient drug against Plasmodium falciparum, the reactive agent of malaria. Malaria affects mostly people in the tropical and subtropical regions and caused 409,000 deaths and an estimated 229 million cases in 2019 alone. To follow in the footsteps of the iGEM team Hamburg 2018, which was working on a different approach to fight malaria, we selected artemisinic acid, which can be chemically transformed to artemisinin as one of our target products. Biotechnological production of this pathway, identifying CYP71AV1 as a key enzyme, has been established in E. coli strains, and we wanted to continue this impressive work. Our approach would use plasmids designed by Kiesling and co-workers, which encode for the required enzymes to establish the mevalonate pathway in E. coli, in combination with the FPP and amorphodiene synthase, as well as CYP71AV1 to catalyze the crucial functionalization of amorphodiene to artemisinic acid. We envisioned further increases in the product titers of artemisinic acid to enable new perspectives for the production of semisynthetic artemisinin. The production pathway for artemisinin is a good example for a dual synthesis strategy, using both microbial fermentation as well as classical chemical synthesis. By combining these different approaches, the potential for optimizations is maximized. Another sesquiterpene lactone which attracted our attention is pathanolide, which exhibits a broad spectrum of biological activities like anti-cancer effects. From a biosynthetic point of view, pathanolide is derived from the terpene germacrene A, which itself is a cyclization product of FPP by the corresponding terpene synthase. The biosynthesis of pathanolide occurs by several CYP enzymes, which makes implementing this pathway much more challenging. Complete synthesis of pathanolide has not been shown before in E. coli. A third idea we had was to produce green platform chemicals, which could be used as biofuels. The CYP enzyme OLE-T decarboxylates a broad scope of carboxylic acids to the corresponding terminal alkenes, which can be used in the petrochemical industry. Although the fusion of OLE-T with the CPR is already described in literature, we wanted to include OLE-T in our toolbox for designing CYP-CPR fusion proteins to demonstrate the general applicability of this approach outside of terpene biosynthesis. Most of our research focuses on E. coli, but the use of phototrophic organisms for chemical production applications appears to be particularly promising and sustainable. Cyanobacteria, for example, can produce the desired products like terpenes directly from light and atmospheric carbon dioxide. By using the electron carrier ferredoxin as some kind of linker between CYP and photosystem 1, it should be possible to harness electrons directly from the thylakoid membrane. This could enable a more precise control of the flow of reduction equivalents, so that the desired reaction does not directly compete with other metabolic processes and effectiveness of the fermentation process is increased. Complementing our project are ideas to modify the energy metabolism of the microorganism to better allow the regeneration of reducing equivalents like NADPH, which are needed for catalyzing the CYP enzyme reaction. Therefore, we plan to additionally express glucose 1-dehydrogenase 2 in the cells to increase the amount of chemical energy available for biosynthesis. During our project planning, we implemented the feedback of different stakeholders to whom we reached out during our human practices work. This led to some changes regarding the target products and construct design. Additionally, we needed to get a lot of expertise regarding the work with cyanobacteria. We then designed our DNA constructs and planned our experiments. On the one hand, we wanted to show that we are able to produce the target products in our bacteria. On the other hand, we designed an activity assay to investigate the effect of the linker sequence on the activity of CYP proteins. We accomplished assembling a large part of our constructs, but unfortunately, the formation of any of our target products could not be detected by gas chromatography-coupled mass spectrometry. There are different explanations for this observation. In general, the functional expression of cytochrome P450 proteins in bacteria can be challenging. Previous studies have shown that only some CYP enzymes can be functionally expressed. For some of our constructs, the expression of N-terminal his-tagged CYP proteins was observed using dot plots, but these results do not indicate whether the proteins are functional or not, and the amount obtained by protein purification was not high enough to further investigate this. Additionally, we struggled with integrating the mevalonate pathway and expressing terpene cyclases in E. coli, leading either to the expression of amorphadiene or humulene. Production of these terpenes has been shown to be possible, but we were unable to replicate the results. We then focused more on approaches guided by bioinformatics for designing a linker sequence connecting CYP enzyme and reductase. We took inspiration from nature, where similar fusion complexes occur, and developed the automated command line software tool LEA, linker extraction from alignments, which is based on multiple sequence alignments. For optimal electron transfer, reaction centers of the proteins need a specific distance, and therefore the proteins cannot be connected arbitrarily. Instead, a linker sequence is inserted between both sequences, allowing a favorable relative position and orientation of the protein domains to make electron transfer more efficient. Finding the optimal linker sequence is not a trivial process, since the 3D conformation of both proteins is unknown. But how were we then able to find a suitable linker? Our software looks at a set of known fusion proteins and calculates linker sequences based on similarities using blast searches and multiple sequence alignments. In the future, we want to include 3D structure prediction of the designed fusion proteins using software like AlphaFold to confirm that the predicted sequences are structurally sensible. In our project, we used this tool to obtain 6 different linker sequences for a CYP1A1 CPR fusion protein. To test if our predictions are correct, we then aim to design a cheap, fast, and reliable assay to assess the functionality of the different fusion constructs. Since the CYP enzymes for terpenoid synthesis require expensive substrates and produce products that are difficult to detect, they are not suitable for this purpose. Therefore, we chose the model enzyme CYP1A1, which catalyzes a reaction which can be easily tracked by fluorescence measurements. With our assay, we could show the rapid conversion of the substrate in both E. coli wild type as well as in modified bacteria transformed with our fusion proteins. But even after optimization, the assay did not achieve the required sensitivity to distinguish between wild type and modified bacteria. Either our constructs were non-functional, possibly due to misfolding during expression, or the assay's signal-to-noise ratio is too low to resolve the difference in activity. To eliminate issues with the assay's sensitivity, an in vitro assay using purified protein would be the logical next step. For this purpose, we added His tags to our proteins, but so far we're unable to obtain sufficient quantities of the purified proteins to develop this assay. In parallel to expressing CYP enzymes in E. coli, we were also working with Cyanobacteria. This posed an additional challenge since we have never used phototrophic organisms before and our lab was initially not equipped to do so. That is why, during all phases of our project, we reached out to various experts in the field to get our project on track. But besides some unsuccessful conjugation experiments, no progress could be made for this part of the project. Unfortunately, despite all our efforts in the lab, positive results showing that some parts of the project are working as expected are still pending. We still have a lot of ideas for improving our project, but time constraints prevented us from exploring all possible options. There are plans to continue the project in the future, hopefully leading to better results. An important part of iGEM is to design your project in a way that it can be built upon by other iGEM teams. This was something very close to our heart. Initially, our first design lacked the necessary modularity for this. We designed the assembly of each construct individually, and the exchanges of parts like promoters was not easily possible. Therefore, we made our Let's Fets collection compatible with modular cloning, utilizing the designated fusion sites and making it possible to use parts from other iGEM collections, like the Marburg collection. In our design, we introduced specific fusion sites for the construction of fusion proteins consisting of cytochrome P450 enzymes and reductases connected by a linker sequence. Additionally, we specified which fusion sites proteins expressed in an operon structure should have. This modification increased the usefulness of our design, and we hope that many iGEM teams in the future will utilize this collection in their project. Before implementing ET, the Electron Transferrestrial, in real life, there is some room for improvements like integrating a secretion system for the produced terpenoids or using a regulatory system based on small regulatory RNAs to allow further metabolic control of this pathway. Additionally, to really be useful for the world, a scale-up process would be necessary, which might pose a challenge. We hope that our iGEM project, which established the mevalonate pathway and production of terpenoids and microorganisms inside the iGEM community, will be an example for many iGEM teams in the future to work on this topic and engineer better, sustainable ways of producing important chemicals used in our everyday life. This will be necessary because we are living in an ever-changing world. Nothing as it is will stay forever, and knowledge is key to transform our society and prepare us for the future. It is important to take fears and misunderstanding in our society seriously. We, as a scientific community, have the obligation to share our knowledge and to show on which principles science is based. We are responsible to strengthen the belief in scientific evidence and educate people of all ages and backgrounds. We wanted to raise awareness for science-related topics and create a safe space for critical discussion. To achieve this, we created an educational program for children with experiments, presentations, and explanatory videos, and hope that this material will be used by future iGEM teams. Our goal was not only to reach people already interested in science, but also everyone else. But how can you fascinate people about something that is foreign to them, that is so strange and not part of their world? We put much thought into this question and found that the most important thing is to interact with people on a personal level, to tell them about science with examples from their lives and reimagine things they love. Together with other iGEM teams, we started a collaboration to look into non-conventional forms of science communication. In a workshop, we came up with strategies for combining art, music, dance, theater, and storytelling with science and presented acts with great success in front of a diverse audience. We wanted to transform complex scientific information into abstract expression without losing the essence of our research. To go even further, we worked together with Philippe Lobos, a Chilean artist, to unite two worlds that could not be more different: science and arts. Using colors, feelings, impressions, art, and audiovisual content, this method of communication allowed us to connect science with feelings. Our goal was to reach people on a deeper level. We wanted to show the passion that we feel for our research and hoped that this would make people more interested in our work. We also invited other iGEM teams to share their creativity and become part of this science meets art collaboration. Now the end of the iGEM season is near, and far too often our innovative iGEM projects discontinued and forgotten. To ensure this won't happen to our project, we need you to continue the journey leading to a sustainable future. Replacing resource-intensive processes for the production of chemicals used in our everyday life with green alternatives relying on microorganisms is a great start. So, be inspired and inspire others. We are the future. Look at the Earth. What a beautiful planet. True, but these humans are destroying her. They don't know what will happen to them. I think they know, but just don't care. Probably true. But there is hope. You heard about the ET project, haven't you? Revolutionizing. But will it be enough?